Happy Thursday, Epidemics. We haven't seen each other in a while. I understand you guys have gotten a lot of snow recently, or at least snow rain mixes. I hope everybody was able to stay home and chill and just kind of relax and have a nice day off. So our announcements as usual. We are going to have assignments posted this week on posted tomorrow that'll be due March 10th. It's really just going to be about today's material. So there's going to be a quiz on today's material, and I am going to have a journal entry up for you guys to fill out. If you haven't submitted your requests for your project yet, those are due tomorrow. And you can submit both the topic and the format on Blackboard. If I give you a grade of 1 on the assignment, that means it's approved, no problem. Just go right ahead, have at it. If I give you a 0.5, that means I left you a comment. If I gave you a 0, that usually means either I think the topic's going to be very difficult to do a project on, or more likely somebody else has already signed up for that one. And spring break is coming up fast. So we have meetings next week, and then there's a week off. All right, another week off. Okay. So what we're going to be talking about today is vaccination. Last time we met, we were talking about how the immune system functions. And, you know, it's a complicated topic. The immune system is incredibly complicated. The vaccines, we're going to talk about different kinds of vaccines, how they work, and just kind of an overview of it. They are actually just as complicated as the immune system, but we're not going to go into that much depth on this topic. So a vaccine is something hang on, here we go. A vaccine exposes the immune system to antigens. Remember, antigens are anything that stimulates an immune response. And they do that so that the memory cells will recognize that antigen if it sees them again. The goal here is to prevent the infectious, the pathogen from becoming established. So I say from becoming established because we're not preventing an infection. What we're doing is we're preventing it from becoming severe. So it's more, reduces the severity. And a good vaccine with a healthy immune system can often reduce the severity to the point where you don't even know you got the disease. You don't have any symptoms. You don't have any disease process going on. Basically, the immune system comes in, recognizes those antigens, wipes them out, and that bacteria or virus cannot cause the disease. So there are several different ways we can make vaccines. And I've got a little list here on the bottom. This is not a complete list. And figuring out which type of vaccine to make is very, very complicated. So we're going to go through these 4 major categories. The first one is called live attenuated. So attenuated is a scientific word that means that the bacteria or the virus has been modified so that it is unable to cause an infection, or at least not a severe infection. So you have live virus or live bacteria. This is not safe for people who are immunocompromised because it does require your immune system to mount a response and kind of get rid of that attenuated virus or bacteria. The advantage to live attenuated vaccines is that they produce a very long-lasting, very strong immune response. Your immune system recognizes that this is a live organism. I know viruses aren't alive, but a functional virus or a live bacteria. And so it's going to kick in pretty strong and be saying, okay, we have to knock this out fast. Examples of live attenuated vaccines include the measles, mumps, rubella vaccine and the chickenpox vaccine. I should also mention that the polio vaccine is a live attenuated vaccine. So with the MMR, measles, mumps, rubella, and chickenpox vaccines, those are safe as long as you're not immunocompromised. The polio vaccine does sometimes cause cases of polio. It's usually a milder disease than the naturally occurring polio, but it can cause disease and it can cause harm in people. Polio it's a disease that we've almost completely wiped out. People who get the symptoms, it's a nasty, nasty disease, and it can cause paralysis that can be lifelong. And it can even cause paralysis of the muscles involved in respiration. We're going to see a picture later on that shows how we had to deal with polio back when it was a big problem. So that's live attenuated. The next category is killed whole cell. What this means is they grow up the pathogen, virus or bacteria, and then they kill it. They make it, well, with viruses, this is we call it killed when it is unable to cause any kind of infection. So they use radiation or chemicals to kill it off. These cannot replicate. The live attenuated pathogens can replicate. The killed whole cell pathogens cannot. These don't last as long. They often require multiple shots. And right now, what we use this for are flu and cholera. So flu, we have to get new shots every year, not only because the vaccine does not last very long, but also because the strain of flu changes from year to year. And we're getting ready to talk about the 1918 flu pandemic. So we're going to get into the flu and we'll talk about how it changes and how it modifies. Our third category, I kind of lump these 3 together: component, conjugate, and toxoid. There are subtle differences between these 3, but they all take part of the pathogen or part of a toxin produced and use that as the vaccine. These are very, very, very safe. There's no cell, there's no virus. It's just part of the pathogen that's injected. And so with this one, we can think of tetanus. The tetanus vaccine, we put a little bit of the toxin from tetanus, just a smidge of it, so that the immune system will recognize it, but not enough that it actually is functional as a toxin. And our fourth category are the new ones. These are the mRNA vaccines. This one is similar to the killed whole cell in that we're only going to be making an antigen. We're not going to be making a bacteria or a virus. We're just looking to get that antigen produced. And the way we do this is really pretty cool. We take, so let me step back. Our genetic material is DNA. And in order to make proteins from that DNA, we have to convert that DNA first into mRNA. The M stands for messenger. And that mRNA can leave the cell nucleus and go to the site of protein manufacturing. And then it has the instructions for how to make that protein. So with mRNA vaccines, we're injecting that messenger RNA into your cells, and your cells now make the antigen. So your cells are manufacturing that antigen. The immune system can then see that antigen, respond to it, remember it. This is very, very new technology. The COVID vaccine, as far as I know, was the first one to be manufactured with this technology. It's very safe. It's easy to modify. So you may be aware that we have a different vaccine now for COVID than we did at the beginning of the pandemic. There's a lot of talk about using this to make flu vaccine that's a little bit more accurate, because the process of making the flu vaccine takes a long period of time. And so researchers look at all of the strains of flu that are going around and make an educated guess about which strain is going to be circulating months down the line. With an mRNA vaccine, we can theoretically ramp up production much closer to the time when we tend to have flu outbreaks, and then we're much more likely to get the correct strain of the flu to fight against that correct strain. Now, vaccines are not permanent, and different vaccines work for different lengths of time. Some vaccines only work for 1 to 2 years, like the flu vaccine, for example. In fact, flu shots usually only last a few months in an individual. But since flu is not something that happens year-round, it's something that happens within a specific time period, that's okay, because you only need to be protected during flu season. Others will last for longer. The measles vaccine lasts for decades. Depending on what type of vaccine you received originally, you may need to get a booster in early adulthood. I had to get a booster in early adulthood. But not everybody needs it, because different measles vaccines are turning out to have slightly different survival periods, or your, let me back that up. Your memory cells remember the different measles vaccines for different lengths of time. But regardless, it's going to last for decades. Measles is another disease that we've largely eradicated from our society, I should say. So we don't really understand how very, very deadly this disease was. Some vaccines require multiple doses, and these may need to be spaced out in time. The shingles vaccine for older adults needs 2 shots. Rabies vaccine requires 3 shots. HPV vaccine, I think they're still requiring 2 or 3 shots for that one. So how many you need and how spaced out they are, this is all stuff that has to be figured out by the vaccine developers. So you can, hopefully you're getting the idea that this is pretty darn complicated. And vaccines may need to be given at different ages. So this is for 2 reasons. Some diseases are more likely to infect certain age groups, or more likely to be severe or deadly in certain age groups. Sometimes it's the immune system that needs to be more mature. So very, very young children, the immune system's not very mature for the first few months, first couple of, up to probably it's not really mature until probably around 2 years of age. But so depending on the vaccine, you may need to wait until a child is a certain age because their immune system is not developed enough to mount a good response and to have a good memory of it. The older you get, then your immune system starts to decline, and you may require boosters of things that you've already been vaccinated for when you were younger. And then with the age period, we can look at something like meningococcus B. The meningococcus diseases, or meningitis, these are diseases that infect the brain, specifically a specific part of the brain, but it is infecting the brain. It can cause swelling of the brain, and your brain's inside of your skull. So if it swells, there's nowhere for it to swell to. It's going to create a high pressure within the brain itself, which can cause permanent neurological damage. It can cause death. And some of these meningitis diseases can cause death within 24 to 48 hours of symptoms. Meningitis, meningococcus B specifically, is a concern primarily for people between the ages of 19 and 23. I don't know why this is the case, but this particular age range is very vulnerable to this disease. College students. Let's talk about side effects of vaccination. What kind of side effects have you seen yourself from vaccinations? Does anybody want to chime in?
Muscle fatigue.
Muscle fatigue, yeah, that's a good one.
Definitely like getting really tired.
Getting tired, yes. Those are the 2 biggest ones. Fatigue, a sore arm, muscles, or even a mild fever. These are all very, very common side effects from vaccines. The COVID vaccine in some individuals can cause stronger side effects, especially mild fevers. Severe, serious side effects are very rare. If you think that you have been harmed by a vaccine, if you think that you've gotten some severe side effects, you can report your side effects to this organization called VAERS, the Vaccine Adverse Event Reporting System. This is run by the federal government. Anybody can report. So this means that if you think you've gotten a side effect from a vaccine, you can report it. The idea here is by having individuals reporting side effects, we're more likely to detect major problems with vaccines early, rather than waiting for people to tell their doctors and then the doctors to report it. And yeah, you know, that's going to be a much slower process. Now, the problem with VAERS is that anybody can report on it. And so we get all kinds of anti-vax crazy stuff posted to VAERS. I can go on and say, I got the new bivalent COVID vaccine and it made my skin turn blue. And it'll stay up on the VAERS site. It'll be up there. It's not gonna be taken down. Nobody's, at least as far as I know, nobody's really monitoring for crazy people putting stuff up. So if you're going and you're, you know, not convinced that vaccines are a good thing, you can go to this site and get really, really scared and figure out, okay, I don't want a vaccine because look at all these horrible things that can happen from it. But I want to also mention here that in the US, there is Vaccine Injury Fund. It's a national program that compensates people who get serious vaccine injuries, either temporary or permanent. Most of the serious side effects are severe allergic reactions. These are very, very, very rare. This fund requires proof that you've got a vaccine and that whatever your injury that you're reporting, whatever that injury is, that you actually had it. So a doctor has to go in and say, yes, this person had temporary paralysis, or yes, this person had anaphylactic shock. Between 2006 and 2021, there were over 4 trillion vaccine doses given in the US. That is such a freaking huge number that people can't, unless you work with massively huge numbers, this number just doesn't even make sense. We have right now, the population of planet Earth is somewhere between 7 and 8 billion. And a trillion is like 1,000 billions. So this is a massively, massively huge number. There have been compensations during this time. There have been 7,215 compensations. That should give you an idea of how rare serious injuries from vaccines occur. And the fund is very, very generous. If you can show you got vaccinated and you can show you had an injury, they're going to give you money. They're not going to fight this through the court systems. This is designed to compensate people who are injured. and I took one of my favorite slides out. What have you heard of as major risks with vaccination, especially vaccination of young children?
So the biggest one that comes to mind, which I know has been proven to be untrue, is that vaccine, like certain vaccines in young children, will cause autism, right?
Yeah.
Yeah. So
Yeah. Yeah, and you're right. That has absolutely been shown not to be true. So this is one of those things that frustrates me to death, because this idea came about when an article was published. I didn't write his name down. I usually can remember his name off the top of my head, so I didn't put it in my notes. Wakefield. So there was a British doctor, Dr. Wakefield, who, in addition to being a practicing doctor, also got hired by a law firm. This law firm hired him to look at causes of autism. Specifically, the lawyers actually specifically said, can we say that it's related to vaccination? And the reason they said this was because the age of onset of autism symptoms is about the same age as when children get certain vaccines. And Dr. Wakefield goes, I've been trying to develop a new measles vaccine anyway. Let's go after the MMR vaccine. You may think I'm exaggerating here. This may not have been as straightforward as that, but basically that's what he did. So he gathered a bunch of data, published an article saying, I've found all of these children who have all of these symptoms and signs of autism, and they got autism right after they had the MMR vaccine, and I'm confident that the measles, mumps, rubella vaccine caused the autism. Well, he was lying. The data in his paper was made up. When people tried to repeat his experiments, they couldn't. They couldn't get the same results. And so then they started looking into the patients that he mentioned. And you don't mention patients by name in an article, but you can, if you're an investigator, ask to get the doctor's notes and confirm that those patients really existed. Yeah, well, the patients seemed to have existed, but none of them were autistic. So he just kind of randomly picked children and put them in his paper to say that the vaccine caused the autism. And there are people who still believe it today. Dr. Wakefield is now no longer a doctor. He's now Mr. He's had his medical license revoked. He is currently, he left England because of the scandal. There have been lots of lawsuits against him. He is now living in Texas. He's not legally allowed to practice medicine, but he's still spreading misinformation. The thing that pisses me off so much about this, besides the fact that we've got people who are not getting their children vaccinated, and kids die from measles. Kids die way too frequently from measles. So this is there have been kids who have died because of this. But it's also taken money away from autism research. We've had billions of dollars spent to demonstrate that the measles, mumps rubella vaccine does not have any relationship with autism. It does not cause autism. Every study except for Wakefield's has shown this, and people still believe it. I would rather we have actually figured out how to help people with autism. We have some clues now about what does cause autism. And that is things like fathers at an older age, premature births, very low birth rate or birth weight, other genetic conditions. I'm going to move this over here so I can keep typing. Parental exposure to air pollution. Or prenatal, I should say. No, parental. There we go. The parents being exposed to air pollution, parental exposure to certain pesticides, maternal diabetes, or immune system disorders, and birth difficulties that result in low oxygen to the baby during birth. Whew. That's a lot of different things that we've shown are risk factors for autism. None of them are vaccines. People talk about supporting or boosting your immune system. Well, you can support your immune system by exercising regularly and eating more plant products. This is good. It helps your immune system stay healthy and functional. But here's the thing. There are all these people out there talking about buy this, buy that, boost your immune system. I do not want my immune system boosted because an overactive immune system causes autoimmune diseases. So these are things like rheumatoid arthritis. You can see the person's hands over here and how these joints are badly bent. They've also got some psoriasis on the skin. This is autoimmune right here. There's also muscular dystrophy, lupus, celiac disease, type 1 diabetes, pernicious anemia. Autoimmune diseases are nasty, nasty diseases. They can cause a lot of damage to your body. There is nothing you can take that will boost your immune system. There's no pill, no specific food that has been found to do this. And like I'm saying here, you really don't want to do that. You want a healthy immune system. Healthy immune system, eating plant products, exercising regularly. There's some evidence that having furry pets when you're a child can also help train your immune system. That's some evidence. So what happens if we don't get vaccinated? So you can catch one of these diseases. This guy on the left was one of the, he's one of 2 people in the US who is still living in one of these structures. This is an iron lung. He has been in this iron lung since he was a child, or he's been in a iron lung since he was a child. He's paralyzed and completely unable to breathe. So he has to stay in this structure in order to breathe. This machine changes the pressure inside the chamber there. And so it will, by reducing the pressure inside the chamber, that will cause his lungs to expand, and he inhales, and then it increases pressure, which pushes his lungs in so that he exhales. This is the only way he can breathe. We have other techniques to treat people with polio nowadays, but for whatever reason, there are two people who've decided to stick with this old technology. So polio, you have a risk of paralysis. Measles used to be the #1 cause of blindness. It can cause neurological problems. It can impair the immune system. So measles is one of those diseases where if a child gets measles and recovers, he still has an increased risk of death for the next 2 years because his immune system has been damaged so badly that he's more likely to get an infection, and he's more likely to get a severe infection. And then if we look over here, we can look at the really baddie, death. So this is the global childhood mortality rates. The left-hand bar here, pre-vaccine death rate due to infectious diseases for people before the age of 15. Over 45% of people died of infectious diseases before they turned 15, before we had vaccines. Nowadays, that death rate is about 4%. Pre-vaccine death rate before 1 year of age due to infectious disease. Before vaccines, it was 27%. Now it's more like 2 or 3%. So there's been a huge, huge change in the mortality. Even with this, there are over 4 million deaths worldwide from vaccine preventable diseases. Some of this is from people who are anti-vax. Some of this is from people who don't have the option. They live in an impoverished country or someplace where it's very hard to get vaccines. Oh, here we go. No, damn it, vaccines do not cause autism. Measles and mumps. The anti-vax movement in the US really started surging in 2007. And since then, there have been 9,000 vaccine preventable deaths in the US alone, and 150,000 preventable disease infections. This poor child right here is an example of what measles looked like. I guess still does, because it still exists. There is a condition called Guillain-Barré syndrome that has a small, it's been associated with vaccines, and there is a small risk with certain vaccines, specifically the meningitis vaccines, which Guillain-Barré is a condition of the peripheral nerves. So these are the nerves that are not part of the brain or spinal cord. They're in the rest of the body. So there is a risk of paralysis. There is a risk of death. Most cases of Guillain-Barré, we don't know what caused them. This is called idiopathic. We do know that flu infections can cause Guillain-Barré. And Guillain-Barré can happen at any age. to pretty much anybody. So while there is a small risk, it's not as big of a risk as catching some of these nasty diseases that we could prevent with vaccines. I want to mention a couple of these diseases specifically. I want to mention whooping cough. Whooping cough is a fatal disease for the very, very young and the very old. It can infect anybody at any age, and healthy young adults can get this and it can cause coughing that is so severe that it can break ribs. It can cause loss of bladder control. It can cause vomiting, and it can last for months. I have had a student actually who took this class a couple of years ago who did catch whooping cough. College-age person, young and healthy. She had whooping cough that lasted for months. Rabies. Rabies, unvaccinated, there's a 100% death rate. Now I'm going to put a little asterisk here because there have been roughly 5 people who have survived rabies without the vaccine. Most of those have been in the past few years. There's a new treatment plan that seems to help in very rare cases, but it's a long, difficult treatment plan. The good thing about rabies is if you get exposed, you can be vaccinated after the fact. As long as you're vaccinated before you show symptoms, you're good to go. So if you get bitten by a wild animal or a stray dog or a stray cat, or you wake up and find a bat in your bedroom, go get your rabies boost vaccine. It's not hard. It's not that painful. I've gotten rabies vaccinated, and it's 3 shots, which is annoying, but it's shots in the arm. It's not shots in the stomach. I know there's a rumor that you get these vaccinations in the stomach, but no, it's not. And we have a vaccine for smallpox. You guys know this. We know we've eliminated the smallpox disease. It's been wiped out. But I want to remind you about this because it does still exist in freezers. And there is a potential for bioterrorism. So remember, half the people who are exposed get smallpox. Those who are exposed, 1/3 of them die. So that means 15% of people who are exposed to smallpox will die. Smallpox is airborne up to at least 6 feet. Vaccine immunity, we have no idea how long this vaccine lasts. The estimates range between 6 and 30 years. So that's a very long range. Routine vaccination against smallpox in the US ended in 1972. So that's more than 30 years ago. So even those of us who got vaccinated for smallpox, unless you're in the military and you've gotten it recently, yeah, there's hardly anybody immune at this point. Okay, so that is our discussion of vaccination. What kind of questions do you guys have? Okay. So I'm going to send you guys out into a breakout group. Where is my? Why am I not seeing the breakout groups? Okay. I'm just going to put you into 2 breakout rooms, and I want you to talk about the anti-vax movement. Why are people so hesitant to get vaccinated? Do you think that we have lost the understanding of how severe these diseases are? Do we not, as a society, do we not trust science? Do we not trust doctors? What's going on that has caused this surge in anti-vaccine ideas and rhetoric? And is this a problem? Should people be allowed to make those decisions for themselves, even though not getting vaccinated means you're more likely to catch a disease and then potentially spread it to somebody else? Okay, and I'm going to give you 5 minutes. Okay, everybody, I'm getting ready to close these rooms. You have a minute to wrap up your conversations. Okay. So what did you guys talk about?
So my group, we kind of talked about how like social media can definitely make it worse for people who don't get vaccinated. But we definitely we understand both sides. Like my group, like we're pro-vaccine, we get vaccines and we're here for it, but we also understand like why people don't get them because, you know, some people actually are terrified of what they're putting in their bodies and then they see on social media and then it gets even worse for them. Like, so we understand why, and especially like with the COVID vaccine, we get how some people didn't get it because it was like pretty new and they wanted to like see how it was first and then, you know, maybe decide to get it or not. So yeah, we just said that social media just really makes it worse. And we think people should have a choice to get a vaccine or not. Like there shouldn't be a law to make it like prime, like a law to like get a vaccine.
Okay. What about the other group?
Well, I was doing a bit of research during our group. I noticed that some of the least likely to be vaccinated were like ethnic minority groups.
Mhm.
And I mean, when you take a look at government's history in the US, we don't exactly have the best track record with stuff like forced sterilization, stuff like that. So understandable why they might be a bit reluctant.
Yes, yes, absolutely. That is definitely a big problem with minority people and medicine. They don't trust medicine, and for good reason. Anybody else have an opinion they want to chime in with? Okay. In that case, we're going to be moving on to our next disease, flu. Specifically, we're going to talk about the 1918 flu pandemic. So I'm going to read you a couple of quotes here. The first one is from a woman named Josie Mabel Brown. She was a nurse at the Great Lakes Naval Station during the 1918 pandemic, and this story was told by her niece. Quote, as she walked into the ward, not only were the 42 beds full, but there were boys that were laying on the floor and on the stretchers, waiting for that boy in the bed to die. They had raging fevers and delirium and profuse nosebleeds, and their lungs would collapse and it would go into this horrid, bloody pneumonia. Second quote is from Arthur Davis, a gravedigger. One morning at 6 a.m., I was set to work digging 3 graves for a family of 6 that lived down the road from my home. Around 9 a.m., the doctor sent word to dig yet another grave. Then around lunchtime, I got word to dig yet another grave. And by 4 p.m., I was instructed to dig the final grave for that entire family. So this was not the flu as we know it today. It was a much, much deadlier disease back in 1918. So we're going to do a quick look at the numbers here. The Black Death, which we'll talk about after spring break, is hands down the deadliest pandemic ever. It is estimated that between 25% and 30% of the world population died within a small number of years. This was 75 to 200 million deaths. The American plagues, this was a series of different diseases that were brought to the Americas when the Europeans invaded. It is estimated that up to 90% of the indigenous Americans who were living in the Americas in 1490 died. In Mexico in 1500, the population was around 21.5 million. 100 years later, it was 1.5 million. Columbus landed on Hispaniola in 1492, to give you an idea of the time we're talking about here. The 1918 flu killed around 3% of the world population, 50 million worldwide deaths. I have not updated the COVID numbers on this slide. Apologize for that. We're definitely well past 25% of the population has been infected. And we're at a little more than 1 in every 300 Americans that have died of COVID in the past 3 years. We are definitely somewhere more than 1 million deaths in the US. Last estimate I saw was about 1.6. We're not doing a good job of keeping track of this. And that doesn't take into consideration long COVID, which is disabling for months, years. We're not sure how long. So regardless, we're definitely well below 1% of the population dying of COVID. One of the problems with the flu is that severe cases of the common cold, some other viral infections, people call it the flu because they want to say, look, this is bad. It's not just a little cold, it's a bad cold. So they call it the flu, but this leads to a misunderstanding of how severe and how serious the flu can be. It's an easy mistake to make because the symptoms of the flu are very similar to other respiratory diseases. So we get generally nausea, fever, fatigue, vomiting, lasting for 1 to 2 weeks. So in most years in the US, flu kills 20 to 60, in a bad year, 100,000 Americans. So it still does kill a lot of people. But the 1918 flu was even worse. The modern Western medicine was just starting to understand infections. In the 1800s, the study of bacteria was just starting. We had microscopes, we could finally see these little things. Germ theory still took a while. We knew before that diseases could be spread from person to person, but the idea that bacteria caused disease didn't start until the late 1800s. The two big names that really worked on this idea were Pasteur and Koch, and they did work in the 1860s and 1890s. We will talk about those a little bit later, especially Koch's postulates. We developed the first antibiotics in 1928. So this was a decade after the 1918 flu. They wouldn't work on the flu because the flu's a virus. Antibiotics were not used on a person until 1941. it's what happened to discover this is Alexander Fleming noticed that bacteria would not grow near mold contamination on his petri dishes. So the bacteria stayed away from the mold. It was very, very, very hard to extract the penicillin though. So efforts began in 1939. The first patient who was treated in 1941 and used up pretty much the entire supply of penicillin in the United States. The entire supply, and died anyway. He had a remission, he felt better for a while, but we didn't have enough penicillin to get rid of the infection completely. Before antibiotics, it was very common to develop life-threatening infections, even from minor injuries. One of the last major wars before an understanding of germ theory was the Crimean War in 1854 to 1856. 34,000 soldiers died in action, i.e. they were killed by the war. 130,000 died from diseases. So that's 4 times as many as died from actual war injuries, events, whatever. The first virus wasn't identified until 1892. Viruses, it was known that they existed, but it wasn't known what they were or how different they were from bacteria. They were called filterable because they would pass through a filter that would stop bacteria. So if you poured a culture of bacteria across this filter, the bacteria would not be able to get through it, that filter, but the viruses could. So we knew that something existed, but we really did not understand what it was. In 1892, the tobacco mosaic virus was identified. It's one of the largest viruses in the world. So even at this point, germ theory was still relatively new. Not everybody believed in germ theory. And a lot of people did not trust doctors, more so than now. One of the reasons for this was it was very easy to get into medical school as long as you had money. You didn't have to have good grades. You could get a degree, become a doctor, and not know the first thing about how to treat diseases. And the flu virus itself wasn't identified until the 1930s. So we didn't know what caused the flu during the 1918 flu. And treatment was pretty much limited to bed rest. We didn't know how to treat it. So this was what was going on during this time period. Very early in our understanding of disease, very early in our understanding of how to treat disease, no treatments for, no antibiotics, very few vaccines, and here comes the 1918 flu. So, since we didn't know what the flu was in 1918, how do we know that all of those deaths were caused by the flu? Well, in the late 1990s, scientists were given permission to go to Alaska, where 72 of the small villages, 80 adult inhabitants, were buried after succumbing to the 1918 flu. 72 out of 80 adults died in the 1918 flu. This is the graveyard. The reason the researchers went to Alaska is because the bodies were buried in permafrost, so they were well preserved. The virus is still there. They isolated the virus, they sequenced the virus, and found the flu. Another team also got flu virus from preserved lung tissue from a 21-year-old US service member who died of the flu in September of 1918. And yet a third group got samples from a 30-year-old service member who died of the flu. None of these samples was complete. The virus is not a complete virus that's capable of infecting. We don't have to worry about melting permafrost, bodies having preserved flu virus. It's not preserved. It's broken down. So because they didn't get a complete sequence from any one of these, they still were able to take these samples, multiple samples from this gravesite, from these two service members who we had tissue samples from, and we were able to combine the sequences to get a complete sequence of the virus. Now, I've been using sequencing and sequence. What do I mean when I talk about sequencing? No idea, huh? Okay. So when I say sequence, I mean the genetic sequence. So we can read all of the genetic code from these viruses. This means that we can confirm that it is the 1918, or that it is a flu virus, and we can look and try and figure out what exactly made this year so deadly. To try and figure out what was going on, scientists recreated the virus, infected cells in culture, and eventually infected mice. One researcher worked with this virus once it was recreated. He worked under what's called enhanced ESL3 conditions. So this is everything is done inside of a hood, which has special ventilation to try and prevent the virus from getting out, wearing the bunny suit and the gloves and the booties. He probably had a respirator to protect his face. So very strict safety precautions. There were retinal scanners to prevent anybody else from getting in. This researcher took prophylactic or preventative antivirals that do work against the flu nowadays. And if he got infected, he would be placed in solitary quarantine without access to the outside world. So that's not spelled right. So he would not have nurses and doctors coming in to treat him. Basically, it's here you go, you're in quarantine, you're not going anywhere. So he volunteered to risk his life to try and understand this 1918 flu. but they worked very, very hard to make sure it didn't get out. What they found was 1918 flu replicated faster than other strains. It produced more additional virus faster. It also spread rapidly in the lungs of the mice. This rapid and severe lung damage was common during the 1918 flu. It was something that was seen in a lot of these, lot of patients. And it was at least 100 times more deadly than modern flu strains. They tried modifying specific genes in the genetic sequence to see if they could figure out which gene was doing it. And it turns out that any of the genes that were involved in copying the genetic information of the virus in that replication process, the virus was milder. If you replaced the surface proteins that help the virus attach to cells and infect cells, the virus was less deadly. So it was a combination of genes that made this particular strain so very, very deadly. Okay, and we're going to have to stop there. Anybody have any questions or comments before we break apart?
All set.
All set?
All set.
Okay, then I will see you guys next Tuesday, as long as you don't get another snowstorm.
Have a great weekend.
All right, thank you.
Bye.
Bye. Thank you.